Imagine you are trying to find a microscopic trace of a banned pesticide on a commercial farm. If you take a swab from one of those plants, you aren't picking up a clean sample of that pesticide. You are scooping up a chaotic chemical soup containing thousands of totally harmless organic compounds, plant waxes, sugars, and soil nutrients. Normally, chemists use a mass spectrometer to identify compounds. Think of it as a highly precise molecular scale that measures the mass of particles. But that creates a problem. In a complex biological soup, dozens of completely different molecules might happen to weigh exactly the same amount. When you run that raw sample, the data for those identical weights gets piled on top of each other. The readout becomes a cluttered mess, and the tiny signal of the pesticide you're looking for is completely buried in the background noise. Simply weighing the sample is insufficient. To prove the pesticide is actually there, scientists have to figure out a way to physically pluck that specific molecule out of the surrounding noise. Historically, the solution was to rely on physical separation methods, like chromatography. This involves forcing a liquid or gas through a long column, teasing the chemical mixture apart over a span of minutes or even hours. But in fields like forensics, anti-doping, or rapid food safety checks, waiting hours isn't an option. You need a definitive answer immediately. That is where Tandem Mass Spectrometry, or MSMS, comes in. Instead of separating the chemicals slowly on a laboratory bench, it uses magnetic and electric fields to isolate them instantaneously, deep inside a sealed vacuum. It condenses a massive, time-consuming preparation workflow into a millisecond-long operation happening mid-flight. Tandem Mass Spectrometry links analyzers in a sequence, like a triple quadrupole. When the sample is shot in, it hits the first quadrupole. This stage acts as an electromagnetic bouncer, tuned to only let particles of one specific mass pass through, say our target pesticide. Every other molecule in that biological soup is instantly destabilized by the electric field. They spiral off course and crash into the metal rods, permanently filtered out of the flight path. The surviving target molecule, now known as the precursor ion, sails straight through the first filter and into the heart of the machine. They have successfully stripped away the background noise, but we still have a major problem. We know what this particle weighs, but we haven't actually proven its internal structural identity. To figure that out, the precursor ion flies into the middle chamber. This is the interaction cell, and unlike the rest of the vacuum, it is intentionally pumped full of inert argon gas. The precursor ion is moving extremely fast. Hitting the argon molecules is a violent collision. The massive kinetic energy of the crash is immediately converted into internal vibration, stressing the molecule until it literally shatters. These newly formed fragments, which chemists call product ions, then flow out of the collision chamber and into the final quadrupole, where they are individually sorted and weighed. This chart shows why this process is powerful. Two different chemicals, dibutyl phthalate and sulfamethazine, have an identical starting mass of 279. But because every molecule has a unique structure, they break apart along different bonds, producing distinct fragment weights. This predictable pattern of shattered fragments acts as an irrefutable fingerprint of the original target molecule. When you look at the final data, the clarity is startling. Because only our isolated target molecule was allowed into the collision cell to shatter, the readout is completely stripped of the chemical background noise seen in standard mass spectrometry. By filtering out the surrounding background ions, scientists can pinpoint a single molecule hiding inside a complex biological fluid with a level of clarity that a single scan cannot provide. Gathering the weights of those fractured pieces reveals structural details that stay hidden if you only weigh the molecule as a whole. This high-speed filtering enables researchers to sequence massive amino acid chains in proteins and map fragments of DNA. Environmental scientists rely on the technique to monitor the atmosphere, detecting trace amounts of industrial pollutants or toxins in the air and water supplies. And the pharmaceutical industry uses it to rapidly screen thousands of new drug compounds, bypassing the lengthy sample preparation steps that used to bottleneck laboratories. The triple quadrupole operates as a targeted interrogation chamber, demonstrating that sometimes the only way to truly verify what something is to break it apart.